My name is David Kumbrock. I'm the Director of Communications here at the CMT Research Foundation, and I am the host for tonight's Gene Therapies Demystified series. Now, in the first part of our three-part series, we laid the groundwork for understanding gene therapies. So if you haven't seen that, you can still go back and watch. It's on our YouTube channel, it's on our website, and we're going to put the link in the chat so it'll be easy for you to find. Now, don't go watch it right now. But if you're listening tonight and you think, wow, I'd really like to refresh on some of the basics here, it's a great opportunity to explore gene therapies themselves. This second session in the three-part series is going to take a closer look at what it actually takes to bring a gene therapy out of preclinical models and into human study. But first, why the focus on gene therapies for this series to begin with? Well, CMT is a genetic disease, so many believe the ultimate solution will be genetic in nature. With that in mind, CMTRF aggressively funds a gene therapy portfolio, including subtypes like 1A, 1B, 2A, 2E, 1J, 4J, and the RS mutations, as well as others that are in preclinical stages of development. It's against that backdrop that we bring in today's speaker, Dr. Megan Waldrop. Dr. Waldrop will speak about one of the most consequential phases of the research process, clinical trial design. Dr. Waldrop is a pediatric neurologist at Nationwide Children's Hospital and an assistant professor of pediatrics and neurology at The Ohio State University College of Medicine. She's here to talk about an ongoing gene replacement trial for CMT2S, using it as an example to teach us about clinical trial design in gene therapy. Before I turn it over, did want to flag a couple of things. First, we've got some exclusive updates on CMTRF research progress after the presentation that we share here with our webinar audience first. We also plan to announce the final webinar in the Gene Therapies Demystified series and give you the chance to register ahead of time, so you'll want to stick around for those things. Second, Dr. Waldrop also plans to answer questions after the presentation. So if you have any questions during the talk, please use the Q&A function in Zoom, and that will allow us to sort through those questions and get through as many as we can before the top of the hour. With that, I'm happy to present tonight's speaker, Dr. Megan Waldrop.
Good evening, everyone. Thank you, David. I'm really happy to be here today to share the experience that I've had running the trial for CMT2S. So we're going to start today with just a bit of an overview of trial design and how you think about bringing it from the preclinical studies to patients, and then go over a little bit of the data that we've gathered so far in the trial, but really highlighting areas that could perhaps be optimized and done better in the future. So I do have some disclosures. I have received clinical trial support from a few different companies. I have participated on some advisory boards, and I have done a little bit of consulting work for some companies. But first, I want to get started with an overview of CMT2S. So this is a condition where typically the symptoms begin in the first to second year of life. Usually that's when it's presenting because the weakness is kind of what we call the most distal, so furthest away from the body, so kind of in the toes and the feet. And the kiddos that have this condition will have some trouble lifting their feet up, so walking becomes difficult. And that's usually when they're brought to some sort of physician to get the workup started. And unfortunately, this does progress over time. It seems to be slowly progressive, but there can be kind of interspersed periods of more speedy or dramatic decline that can be associated with illness. I'd say anywhere from 50 to 60% or so experience that phenomenon. And unfortunately, eventually they do lose ambulation. And what's particularly stressful to parents is that eventually over time, this can affect the breathing muscles. And that's really due to phrenic nerve involvement, which then affects the diaphragm, which is one of the biggest breathing muscles. And so there's need for respiratory support. This condition, though, is caused by pathogenic variants in a gene that's called IGH-MBP2. This is an autosomal recessive condition, which means that both copies of the gene have to have the abnormal change. And this is a protein, or gene that makes a protein that's pretty widespread throughout the body. It's known for its role in kind of winding or unwinding DNA and RNA. So it really can be involved, and we're not entirely sure how involved, but it can, in theory, be involved in lots of gene expression throughout the whole body. So it presents really as a neuropathy, but it's possible that we don't know the full extent of what these changes in this gene are causing. It's a very rare disease. I'd say I don't think we know the true incidence, but one estimate that's been thrown out is 1 in 100,000 individuals. And there is a spectrum. We're going to focus more today on the milder phenotype, but there is a more severe phenotype associated with these changes, and that's called spinal muscular atrophy with respiratory distress. And this just presents sooner and seems to decline much faster. And although what's been reported is that the first symptom is respiratory distress, now that I've had the opportunity to follow about 20 or so of these patients and speak to many patients around the world and their families, I do think there are some subtle findings that begin also what we call kind of in the length dependent or distal fashion that the CMT version presents as. So I do think there are some very, very subtle distal changes that aren't picked up on because the respiratory distress that occurs first in this kind of subgroup is so dramatic. That's what happens. So usually they're presenting sometime in the first months of life with the significant respiratory issues. They probably will need to be intubated or have very significant respiratory support. They're typically difficult to get off respiratory support and will typically pass away usually within the first year of life if there isn't significant support in terms of like a tracheostomy tube or a feeding tube. There are a couple rare cases out there that I'm aware of where there is a child who had significant respiratory distress very early on, ended up getting a tracheostomy, and then is a young school-age child and is walking. So that's kind of a very rare aspect of the phenotype, though. So I really think of this as like a spectrum with more severe, kind of intermediate, and then less severe. So thinking about this gene, which makes this protein that kind of has a wide spectrum of phenotypes, thinking about how do we bring this to the patients, really the first step is, do we know if this works in animals? So we don't want to put anything into humans unless we have a reasonable idea that it's going to be safe, but also efficacious. And so there was a lot of preclinical work done by a PhD here. Well, she used to work here. She no longer works here, Dr. Catherine Meyer. And luckily, there were 3 different mouse models that were available for her to use to do all of these preclinical studies. And this table is a description of them. But really, it's a very severe phenotype, kind of an in-the-middle phenotype, and then the milder phenotype, which we would think of to be more like the CMT2S population. So we had all of these animals to do very thorough studies in, and very thorough studies were done in all 3 of these models. It was a very large preclinical package. I'm just showing a highlight. I don't have time to go over everything, but this is looking at the most severe mouse model. So she had the gene replacement product that she wanted to see how it worked in the most severe mice. And if you look on the graph that I am showing, the orange represents mice who did not receive treatment, and green is a mouse that did receive one type of treatment, one version that she was testing. And then, sorry, did I mess up? Sorry. So the purple, excuse me, represents the mice that were untreated with empty capsid. And then the red also represents untreated. And then green represents treated. And then that is mainly what we're showing here, that there were these mice who were treated with virus A that had great response to survival. So you're looking at the graph in 0 to 100 in terms of 100% being all surviving. So you want that to be very high. And so you can see with one of the viruses that was tested in the most severe model, they had very good response. And then if you look at the more intermediate model, just looking at times of kind of lifespan, so probability of survival, looking at how their neurons looked, she could also show that in those mouse models, there seemed to be a good response to treatment. And then if you look at the milder mouse model, and this is just looking at how long these mice can hang on a wire. And so usually that is something that a typically or a normal mouse would be able to do for about 60 seconds, let's say. That's what she has on the graph here. Whereas the CMT2S type mouse model would struggle and would not be able to do it nearly as long. And red is representing those who were not treated with the product. And it's really hard to see because they did so well, but actually the green and the orange were two different types of treatment product that she tested, and they actually did as well as the healthy or the typical mice. So all of these studies showed really remarkable and impressive and exciting efficacy that we wanted to move it forward, and she wanted to move it forward into patients. So I'm not showing the safety data today. There wasn't anything that really popped up as significant or concerning, so that you will just have to take my word on that. But that led us to then think, how are we going to get this into patients? So of note, this was studied thinking that the medicine was going to be given through the cerebral spinal fluid. So all of the studies were done in a similar way in the mice. And then just to put a timeline on everything, she reached out to myself and my colleagues in 2019 saying, hey, we have this really great data. How do we get this into patients? So then we have to think about, how do we get it into patients? And so you have to really think about, how are you going to measure the efficacy in patients? And so you need to have validated clinical research tools to be able to do that. And this is a very busy slide, but it's just highlighting that we have been doing research in neuromuscular disease for a while now, and we do have several measures that have been validated in several different conditions and have deemed to be acceptable for clinical research. I also want to highlight that there have been measures and tools that have been developed for CMT as well. And there's even ones for CMT in infants. There's also CMT in peds and CMT in adults. There's also a wealth of information for CMT in general. So yes, we did have those tools, but none had really been specifically tested in individuals with IGH and BP2 related conditions or CMT2S. So the next step is, do you really know the natural history of the course of the condition? Because sometimes we will only be able to show a very subtle change, and that can be very clinically meaningful and significant, but maybe hard to pick up on our tools. So you need to know the natural history of the condition so you can really know which clinical tools and outcome measures you want to use to really improve your chances of showing efficacy. And for IGH and BP2 related conditions and CMT2S, there have been a fair number of cohort studies published, but there's nothing that's been a formal natural history study done. And one is because it's so rare. So a lot of physicians in their career may only see one or two of these individuals. You know, if someone's lucky, maybe five or six, but it requires a lot of collaboration between centers, or perhaps something really as exciting as a trial up and coming, to kind of get everyone to come in and agree to do kind of long-term follow-up so we can really learn the course of the condition. I think what also makes it different for IGH and BP2 is that it seems like there's not discrete genotype-phenotype correlation. I'm aware of several families where they had one child who had CMT2S phenotype, and then they had another child who had the SMARD phenotype. So there's definitely something else going on that we don't know quite yet. But once Dr. Meyer reached out, we got like all hands on deck. We need to get a natural history study going. We need to get this moving so we can determine our outcomes and get this into patients. So we developed the protocol, we sent it through the IRB, we got IRB approval, and we were able to enroll 3 participants through December of 2019 through February of 2020. And the plan was to keep enrolling about one a month, maybe up to two, and do lots of testing to try and find good outcome measures and good biomarkers to really assess the efficacy of the trial. We wanted to look for biomarkers in blood, biomarkers in CSF. We wanted to do diaphragm ultrasounds to measure the size of the diaphragm and see if we could detect change over time. We wanted to do nerve conduction studies to look at the nerve changes over time, and then also functional outcome assessments as measured by our physical therapist. However, unfortunately, we all know that the pandemic then happened, and unfortunately, that shut down the world, and we had to pivot. So, because of the history that infections can significantly precipitate decline in this condition, I didn't feel comfortable really mandating people to travel, but also nobody wanted to travel. So we pivoted to virtual. But that means we lost our blood biomarkers, we lost our CSF biomarkers, we lost the diaphragm ultrasound because it's a very difficult test to do, and there needs to be special training for that. Nerve conduction studies could be done at other sites, but are sometimes done differently. So we did take those when they were offered, but we didn't require it. And then the functional outcome measures, we had to switch to only those that could be done over video. We ended up shipping some equipment to folks around the country and the world to do these assessments over video. So once you have your natural history done, then you should ideally know the best time to plan for your intervention. And that's kind of the next hurdle in trial design. And so unfortunately, we weren't quite sure of that when we started this trial. But prior experience from gene therapy trials and neuromuscular conditions were all kind of pointing towards as early as possible would be better. So we continued on with our natural history study. The vector was completed and ready to go in December of 2020. At that point, we had had 6 participants in our natural history study. We had hoped for 20, but we went to the FDA. We had product. We didn't want to make patients wait any longer. And we assessed their openness to starting the trial with not having great outcome measures selected, not having great plan for timing of dosing, and they let us proceed. So then we had to think about how are we going to design our enrollment criteria. We needed to think about the population that we were aware of. You have to think about, do you want to do a heterogeneous cohort? Do you want to try and do a very homogeneous cohort? And unfortunately, funding and money can play a role in this because if you have a homogeneous cohort, you can do a smaller population. Your N can be lower. If you do heterogeneous, you're going to need to have larger. You could think about, could you do multiple cohorts? Is there going to be an option for expanded or compassionate use? Is that something that could be done? And then you're also limited a bit by the FDA kind of requirements and limitations. So for this trial, they wanted us to start with older participants and then slowly shift the age younger. So at the time of starting this clinical trial, we did not have a good biomarker in blood or CSF. We did not have a complete natural history study, and we had shifted to mostly virtual outcome measures. So we weren't entirely sure which were going to be best for this CMT2S population. We didn't exactly know the best stage of disease, but we thought earlier would be better. But again, we had the limitations set by the FDA. But because this was an ultra rare condition, and I was not entirely sure how many participants would be eligible after screening, the goal was really to try and be as broad as possible. So trying to enroll some with CMT2S, trying to enroll some with the SMART phenotype, and hopefully get a wide range of phenotypes so we could get some good information. So this is the inclusion and exclusion criteria. And so really the inclusion criteria were pretty broad. You just needed to clearly have pathogenic variants. The age range was 2 months to 14 years, and you had to be able to do some outcome measures. Exclusion criteria were, if it wasn't going to be safe to give this product, then obviously we weren't going to move forward with giving the product. And we chose the dosing level to be the same for every participant. We were planning on dosing number of neurons, not necessarily size of the individual. So we really wanted to just focus on targeting the neurons. And typical studies are usually 2 years of follow-up, but because neuropathies can sometimes take longer to see change, we extended it to 3 years of active follow-up and 2 years of kind of the more remote long-term follow-up. And this is a table with the participants in the trial that have been dosed so far. And so you can see we started at age 11 for the first participant and then slowly got younger and younger. And we did 3 with CMT2S at the start. And then most recently, it's been about a year out now since this participant's been dosed. We did another CMT2S participant. One thing to think about in trials is, will the product be seen as foreign when it's given to the child? And so that's where the CRIM positive or CRIM negative status comes into play. So if you're CRIM positive, it's unlikely that your body is going to see the product as foreign, and so you don't have to be as aggressive with immune therapies. For CRIM negative, we added on some additional immunosuppressive agents to try and help minimize that risk. And really, I think the only thing to highlight from this slide is that we didn't have any SAEs related to product. We have seen some CSF protein elevation around the 3 to 6 month time point in a few individuals that we have expanded our analysis on to kind of see why that might be happening. But we have not changed our protocol at all, and the CSF has returned to normal levels, and the participants have not had any adverse effects from this elevation. So really, we have not had any AEs that have led to escalation of care. And by that, I mean adding on additional immunosuppressant or putting them in the hospital or doing other aggressive things. We don't feel like we've seen any AEs that are directly related to the product, though I think jury might still be out a little bit on the CSF protein elevation. We have seen AEs that are related to disease progression, and we did have one participant in the trial who did pass away that we felt was due to disease progression. That child was enrolled right when the respiratory distress was starting, and that once it starts, it's a pretty quick decline. So we don't think that it was related to product. We just think that we weren't able to give the product early enough to stop that aggressive decline. So these are just some of the outcome measures, and the kind of orangey, yellow, pinkish colors are the CMT2S participants, and they're more towards the top because for the functional outcome scores, the number, the better you're doing. And the CMT2S participants were milder in terms of severity. And what we were able to do is we did have some individuals who completed the natural history study prior to dosing in the trial. And so that dashed line in the middle of the graphs kind of shows the quote unquote lead-in data. So when they were in the natural history study, these were their scores. And then once they transitioned into the clinical trial, those were the scores. And so I think you could see for the neuromuscular grow like relative stability, and the North Star assessment as well for the dystrophies. I think you could say, perhaps a little decline or some stability. The 9-hole peg test and the box and blocks. So looking at that, for the 9-hole peg test, it's the amount of time it takes to move these over. So a lower time is better. So increasing numbers represents a decline in function. And for box and blocks, it's the number of blocks that you can move to another side of the box. And so higher numbers is also better there. And so if that declines, then that's less function as well. And I think the most dramatic finding in this one was our first participant had a really significant growth spurt. And then after that growth spurt, there was a significant drop in function. His parents had been really hopeful that perhaps getting this treatment or potential treatment might mitigate any sort of decline that would be seen around the time of kind of pubertal growth spurts. but unfortunately that wasn't the case. The revised upper limb module scores, higher number is better. So they slowly declined over time and for the participants in the trial. And then I think the one thing that is giving me hope is that pulmonary function for the CMT2S participants, aside from participant one and the big growth spurt and some other changes, has been relatively stable and good. And I will say there was one participant in the trial who, over the course of the trial, briefly went on to having a BiPAP requirement. And then at the time, even before the end of the active phase of the trial and continuing through now, has remained off BiPAP. So there's promises of hope, but because we chose to do a very diverse and heterogeneous kind of patient population for dosing, the number is a little too small at this point to tease out definitive signs of efficacy when we're just slowing decline. So we definitely have more work to do. I would say we've always learned and we continue to learn from time to time, which is always really frustrating, that the remarkable outcomes that we see in mice are not always translated to what we see in humans. And then I also do hope that for those in the future, when there are PhDs or basic scientists who are starting to think about bringing a trial to patients and starting to begin their mouse work, they will align with a physician who knows the condition well or has the ability to start concurrently doing a natural history study and getting all of that data that's needed so that we can kind of both be ready and have the best information we need at the same time. Because nobody wants to wait. If there's an available option to try and test in patients and participants, we don't want to have to be waiting years to complete a study and analysis to figure out how to design that trial. One thing that I'm also wondering is the intrathecal or the CSF route of delivery the best way to go? For some of these, the decline is pretty quick, and perhaps intravenous dosing might be another route to consider. So really, we've had issues with funding for this trial, and so we have very limited spots left, potentially only two. I have to go to Nationwide Children's, who's currently funding the trial, and submit a case to request for funding. And the request now for funding has to be somebody who has not been dosed before. And so that really means now we're looking for individuals who are very early symptomatic or pre-symptomatic, so sibling of a family that had a prior child. We've had several people that we've tested, and luckily their current pregnancies have been unaffected. So that's where we are. I'm going to continue to look for funding. We're going to continue to analyze all the data that we have to see if there can be anything really supportive to make the plea for additional funding to continue this trial. So I hope that was a helpful overview, and I'm happy to take any questions.
Thank you so much, Dr. Waldrop. I do have a few announcements I want to go through, and while I do that, I want to invite everyone listening to put your questions in the Q&A section of the webinar that's down at the bottom. We're going to work our way through as many questions as we can. First and foremost, for the announcements, the final webinar in our Gene Therapies Demystified series is going to be called Policy, Access, and the Road Ahead. We're bringing in John Fiore from the Institute for Gene Therapies to examine the federal regulatory landscape surrounding gene therapy development. This is a key area where we need progress in order to get therapies in the hands of patients. It's also a great subject for the community's advocacy if that's something of interest to you. That webinar is going to be July 16th at 7 p.m. Eastern, and the link is going to be in the chat for the registration. Now, I'd encourage you, if you're at all interested, go ahead and register now before you forget, and make sure that you're signed up because it can really help us to plan ahead for those webinars. Second, I want to remind you about the Global CMT Research Convention. That's going to be September 24th through the 26th. It's in Cambridge, Massachusetts, and it's a great event for researchers, but also for patients, families, and supporters. If you do attend, you'll get a chance to hear directly from drug developers, participate in a clinical trial fair to find opportunities to contribute to research directly, and even experience the field's cutting edge technology for yourself, including a motion tracking system that measures CMT symptoms. Not to mention all the great scientific programming that we've got planned for the 2 scientific days of the convention. Now, I want to remind you, early bird registration ends at the end of the month. So if you want to register, you should take a look right now before you forget. That link also going to be in the chat. We also love to keep this group up to date on the progress we're making in research. This month, we've shared key takeaways from a final report on a new way to deliver drugs to nerves in CMT. I'd invite you to go read the takeaways and learn firsthand about the progress we're making. That link is down below. And finally, if you're learning from these webinars, and especially if you're appreciative of the research progress that we're making, we'd encourage you to consider a donation to the foundation. Our highly directed research agenda is supercharged by your support, and it pushes us ever closer to treatments, therapies, and cures for Charcot-Marie-Tooth disease. With that, we're going to move into the Q&A section, and I do have some questions for you, Dr. Waldrop. First, you mentioned a natural history study in the presentation quite a few times. Can you walk us through what a natural history study is, why it matters, and what it accomplishes for those participants in the webinar who might not be as familiar?
Yeah, I'm sorry that I didn't think to do that in my talk at the beginning. Very sorry about that. So when we think about natural history study, we just mean if we were to follow several individuals who had a specific condition over the course of their life, what would the change look like? And it's helpful to describe kind of milestones of change so you have a really good understanding of the disease. Additionally, it's very helpful to know if there are any other components of the disease or the condition that we hadn't necessarily thought about. So, for example, is there also gut involvement? Is there issues with learning or mood? Are there issues with the heart? Is there a high risk for bone fractures or things like that? The natural history can help you determine what to expect, can help you also plan for adverse events and perhaps not be calling something an adverse event related to product when it's actually an adverse event related to the natural course of the condition. And so it just really helps get a full understanding of what you're assessing. So then you can have a better idea of change that might be seen when you're given a product or a therapeutic product.
Got it. One question that comes up kind of frequently in a few different variations is what can we learn from a 2S trial that applies to other CMT subtypes? What lessons can kind of cross apply there, and how does that research intertwine?
Mhm. I think you can learn many things in terms of effect or impact on participants. So every condition that is being studied has different levels of, what's the word I'm looking for? Perhaps abilities or impact on quality of life and day to day. And so you can design your trial to learn how perhaps how much stamina or ability, how many hours in a day could a CMT participant do an outcome measure or do all of the trial assessments. So you can learn, should it be a 1 day visit? Could it be a 2 day visit? You could also learn which outcome measures are perhaps not showing a lot of change and try and reduce, right? So anytime we're doing a trial, we want to minimize the impact and the ask on the participants. It's already a big commitment to participate in a trial, and so we do not want to be doing assessments that aren't going to be high yield in terms of safety or efficacy. So I do think there are certain aspects that can be applied generally across CMT.
Got it. Okay, so CMT2S has a wide range of severity. That's something you mentioned in the presentation, even just within the subtype, right? So, how did that disease spectrum shape who was eligible to enroll in the trial? Is that a challenge other CMT subtypes face also?
I think it could be. So it is an ultra rare condition, so there aren't as many individuals available to participate. So perhaps the varying degrees of severity could impact trial design for another type of CMT. If there is a large patient population, the group who's doing the assessments may decide to focus on one small subset to try and get a good outcome and then expand it.
Hmm.
Um, I think for our trial in particular, there is a big spectrum in terms of either being undernourished or overnourished, and that can play a role in terms of general health for being trial eligible or not. So that's one factor that we had to take into consideration when we were screening participants.
God, I want to follow up on the participant screening. The presentation mentioned that one of the things that you were considering is whether some participants were too symptomatic at the time of treatment. What does that mean in practical terms? But also, how does it change the way researchers are thinking about who to enroll in future trials?
So it's something that has been quite a struggle for me, um, because you walk these journeys with these participants while the trial is being developed. And while the trial is being developed, they unfortunately are having disease progression. And we think about disease severity and symptomatic versus too symptomatic versus symptomatic, thinking about kind of in multiple ways. One is if we do give this experimental product to a very severely affected or very symptomatic participant, do we expect to see change? And so that's, I feel like, the callous part of the researcher. You have to think about the overall results, which is really hard and difficult because you want to think about your patient or your participant that you've been walking this line with. You also have to think about when they become either too symptomatic or more severely symptomatic, does that put them at higher risk for side effects from the potential drug itself or the other things that need to be given with the therapeutic drug? We put these individuals on daily, relatively high dose prednisone, and if they are very tenuously healthy, and an illness could kind of tip them over the edge. That's just something that you have to think about. And it's handled differently, I think, in every trial, which is as I've been giving presentations now about this trial more and more, I've started to think that we just need to build in a cohort or an arm where we just say we are going to dose symptomatic individuals. They're perhaps going to be excluded from our efficacy cohort, or just some way that we can get more individuals who have lived with this and participated in this, just an opportunity to try, even when all the other things are saying that it's too risky or we don't think it's going to help you. Because I think this talk that I just gave is saying, we thought we knew what was going to happen, and we were hoping it was going to be like the treatment for 5-QSMA, and we're having less exciting results. So I still think we have a lot to learn, and it's hard to just say someone's too symptomatic to be in a trial.
God, that's really helpful. Thank you. So we talked about kind of the mix of participants that you're bringing in, but we also often see trials with mixed results. Some patients experience real benefit, others may get nothing. What does it mean for a trial when different participants respond differently? And how, as a researcher, do you sort out what's working from what isn't?
Yeah, that's a great question. I think there are a couple different aspects to think about. So one would be mutation status or mutation type? Or, for example, is there one region of the gene that may perhaps be allow the participant to be a better responder to the treatment versus not? I think for most drugs that have been studied, there are good responders and poor responders. It's just they're in such common diseases that it all kind of fluffs out and looks good even on average just because there's so many. And so when you're in the rare disease space, you really have to think about it as good responders and poor responders. And I think it's on the clinicians to figure out what makes someone a good responder and a poor responder. So then we can continue to move that product forward for those where we're seeing the promising results.
Got it. So we talked about kind of the variance in results there, but there's also the variance between preclinical results and human results. How do you deal with that? What is that gap? Why does that gap appear, and what does it mean to you as a researcher?
When I think about the gap specifically in this trial, in terms of how remarkable the kind of mouse models did with the treatment versus what we're seeing, part of it I think is that mice are small. Their spinal column and spinal cord is very short. And so the drug did not have a long distance to travel. And when you get to human size, obviously the spinal cord is a lot longer. And what we want for this product, so we're giving it in the lumbar space. And so we give the medicine and we tilt the participant's head down in the hopes that the drug will kind of flow up to the top of the spinal cord, but kind of get everywhere. And then we hope that it gets to the nerves and travels down the nerves, all the way to the fingertips and then can have its effect. And so that's a lot of work and a lot of travel. And we know that travel down nerves is very slow and takes a very long time. And so I don't know if the intravenous route is the best route for perhaps somebody who has more progressed disease. I'm still very hopeful in the very early stages of disease where you have a lot of healthy nerve left. It could be a great option.
Got it. I want to drill down on the protocol a little bit there. You had mentioned prednisone. Are there any other immunosuppressants involved in the protocol, or is it just prednisone? Can you talk about that a little bit?
For the participants who we do not anticipate, based on their mutation status, to see the drug product as foreign material, it is just prednisone. So it is oral prednisone, about 1 milligram per kilogram per day, anywhere from 30 to 45 days at that dose, and then we taper. For participants who we call CRIM negative, and we think there might be the opportunity for their body to see this as foreign, we were adding on sirolimus. And so it's an oral medicine. You can give it once or twice a day. It does require some vaccines before giving because it's a more severe immunosuppressant than the prednisone. We did have one participant on that medicine and it seemed to be going okay. It's a medicine that's being used more and more in the gene therapy space.
Got it. And so the prednisone, the immunosuppressant there, is that just part of the trial protocol, or if the treatment were to be approved, would that be part of the treatment protocol as well?
Yes, if the drug were approved, that would be part of the protocol. And I think what's tricky about the sirolimus is the length of time that that stays on would be much longer. And I don't think we've fully nailed down the optimal length of time to remain on that medicine. We're pretty confident about the prednisone. And so we tell, folks it'd be about 2 months on that.
Got it. Okay, so one area that you talked about in your presentation that I always find really interesting is the outcome measures, the tools that are used to track whether a therapy is actually working or not. What makes that so difficult to determine for a rare disease like CMT2S?
I think one is just the small number of participants that you're going to be studying. So if you have a smaller N to your study, you need to have a more dramatic response. Um, when you have, subtleties such as these different variants of CMT, it's, some outcome measures are going to be better at picking up the subtle changes than others in terms of just how that disease progresses over time. There's a push now, and nearly every study that I think every study that I'm aware of has at least one kind of parent or patient or caregiver kind of reported outcome assessment. So we're trying to focus more on not just the strict numbers that we get from PT assessments, but also how is it affecting daily function. And people are also now adding in kind of these activity meters or step counters or other things to try and find out how can we get and measure the actual true meaningful impact for the patients versus just, we came up with this outcome measure. We know that it gets a couple points better. I think we have to be flexible. And I know there's lots of work now in the CMT field to really try and find specific ways to measure change in each of the individual symptoms.
Yeah, that's a really helpful answer. How might lessons from this trial help researchers pick better measures for future CMT trials? As somebody who's, really in it right now, what are you seeing in this study that you think, oh, wow, I hope future trials will pick this up?
I think it's really just time, right? So having the time to investigate as much as you want to investigate to be confident that you're putting the best trial forward. Um, so there have been trials in the past that haven't shown efficacy, and it's always a better feeling for the clinicians to think that we designed the best trial we could. We knew we had the best outcome measures, and we can be confident that this is just not efficacious. And so that's why I mentioned I really hope, and it's going to be one of my kind of platforms trying to move more collaboration between preclinical scientists and clinicians and try and link up early so we can make sure, for example, we have the opportunity to assess all of the different outcome measures I want to think of, to determine which ones to put in the trial. And there are lots of opportunities now. And I think there's now kind of video assessments, AI assisted assessments. I think there are ways to do this without a huge onus on the patients and the participants. Like, I'm not saying we need to design 10 different trials to pick an outcome measure. I think we can use what's out there and analyze it and assess it for our population. We just need the time to do that before the product is available.
Got it. So, for families who are watching this trial and are hoping it might eventually lead to a treatment, what's the most important thing to understand about where things stand right now and what would need to happen for the science to move forward in a meaningful way?
So it would be very helpful to have a participant who's early symptomatic and to get that data. So I think the question becomes, and the question is right now, do we stick with the intrathecal route or the cerebral spinal fluid delivery, and we just need to go a little bit higher on the dose? Or do we need to think about perhaps for those more severely affected individuals, do we need to look at intravenous dosing or through an IV? So there's some data that I'm waiting on that I think will be helpful. And then sadly, of course, we always need funds to do these studies, and everyone knows it's very hard to get funding for conditions that aren't going to be lucrative for companies. But there are mechanisms that I will be exploring when I have those data pieces back and can submit a proposal.
Got it. I want to revisit a couple terms that you've used that I think will be helpful to clear up for our audience. Can you just briefly describe intrathecal and CSF in terms of what that means for a delivery system?
Yeah, so intrathecal means into the space. So if it's similar to when a woman gets an epidural when having a baby. So it's in the lower back, it's the fluid around the spinal cord. It's called the cerebral spinal fluid. and that is the quote unquote intrathecal delivery. So there's a thecal sac that surrounds the cerebral spinal fluid in the cord. And so you're going into that sac and delivering it into the fluid. And then that's the fluid that is along the side of the spine and along the spinal roots that then turn into the nerves that go down to the body.
Got it. That's super helpful. Thank you. Especially the part about the uh, how similar that is to other treatments that are pretty regularly used. That's useful, I think, information. I'm curious how you see the broader landscape of CMT research and where this clinical trial fits into that. How do you perceive the progress the field is making right now?
I think it's really exciting. I mean, to be honest, I'm not an individual living with CMT, so I have a slightly different perspective. I can imagine that it's very frustrating that we haven't made the progress that has been made in other conditions. But I think what's really exciting is that there are now several trials for several different forms of CMT. I remember when it was just CMT1A that was really the only condition being studied. I think one challenge, and to be fair, I am not an expert on every subtype of CMT, but I think one piece that makes this a little bit more challenging is the what I feel like is a very broad picture of how individuals with this condition can look. And I think it's hard to design a trial for just one aspect of it. And so I think that has been one of the more challenging pieces of this as well.
All right, thank you. We are very close to the end of time here, but I did have one final question for you. I'm curious when you're looking at the gene therapy landscape broadly, what gives you hope for the future? What excites you?
Well, I think what's really exciting to me is that more and more conditions have opportunities for gene and genetic therapies. And I think what's particularly exciting are the medicines that work on exon skipping, and they can work on exon skipping. So the exons are kind of like the building blocks of a gene that they stick together and make the protein. And sometimes if one exon is missing, they don't stick together appropriately, and then the protein can't be made. But if you're able to skip one of them, they can fit together then nicely, and they can make almost a perfect protein. And in Duchenne, there are two drugs now that are working really, really well. And I don't see why for other conditions where maybe you could just skip one exon, I don't see why that couldn't be more broadly used. It's very mutation type dependent, so it wouldn't work for everyone, but I think it's another step in the right direction. And those medicines are much less risky than the AAV gene replacement therapies. So I think there are just going to continue to be more and more possibilities. So I'm very excited about that. It's just, the timeline, it takes a little bit longer than everyone would hope.
Yeah, agreed. All right. Well, thank you so much, Dr. Waldrop. This has been really informative, and I hope the audience has enjoyed it too, because I know that I've learned a lot. I do want to remind people that the third part of our Gene Therapies Demystified webinar series is going to cover policy questions regarding to how gene therapies are regulated, and in turn, how we can influence that landscape going forward. Again, that link is in the chat if you want to register right now. You can also find it on our website, cmtrf.org. Again, I'm David Kumbrock, the Director of Communications for the CMT Research Foundation, and I'm so appreciative of everyone who's joined us this evening. And we look forward to making more progress delivering treatments and cures for CMT. Thanks, everybody.